Now that we have discussed how information is stored in the genetic material of cells and how that information is used to build proteins, we need to talk about how that information can be changed or altered through mutation and recombination. A mutation is simply a change in the sequence of nucleotides in the genetic material of an organism. Not all mutations are harmful. In fact, most are neutral and do not alter the structure of a protein. And occasionally, some mutations are even beneficial. Mutations can occur as random mistakes during DNA replication. These are known as spontaneous mutations. Certain chemicals or environmental exposures can increase the probability of mutation. These are known as mutagens. The simplest mutation is a change in a single base pair within a DNA sequence. This is known as a base substitution or point mutation. Because of the degeneracy of the genetic code, meaning more than one codon encodes the same amino acid, point mutations are often neutral and do not change the amino acid sequence of the final protein product. If a point mutation results in a change in the amino acid sequence, it is called a missense mutation. In this example from your book, a cytosine is changed to a thymine. While the pattern of nucleotides in the original piece of DNA encoded a protein consisting of methionine, lysine, phenylalanine, and glycine, the altered DNA generates the amino acid sequence methionine, lysine, phenylalanine, and serine. A single change in the amino acid sequence of a protein can have a major effect on the function of that protein. For example, point mutations are known to cause the important human diseases cystic fibrosis and sickle cell anemia. Similarly, point mutations can affect the function of bacterial proteins. Nonsense mutations occur when the change in the DNA sequence results in a premature stop codon. Here, a T is changed to an A. This changes the codon from one that corresponds to lysine to a stop codon. Instead of producing a fully functional protein, only a partial protein is made. Frame shift mutations occur when nucleotides are deleted or inserted into a DNA sequence. Remember that the ribosome reads the messenger RNA in groups of 3 nucleotides at a time, starting with the first AUG in the sequence. If the nucleotides are added or removed, it can alter the reading frame and completely change the protein sequence. In this example, a single adenine is removed. Now, where the ribosome once would have added phenylalanine to the growing polypeptide, it adds leucine. Each amino acid after the deletion is also changed. Mutations do not occur spontaneously very often, about once every billion replicated base pairs. However, certain chemicals and environmental factors, such as radiation, can increase the rate of mutation. These are known as mutagens. Many chemical mutagens resemble the structure of a normal nitrogenous base and can be incorporated into a new piece of DNA during replication. However, when that piece of DNA is later replicated, the base pairs don't line up correctly, and a base substitution mutation can result. Several forms of radiation can damage DNA. Ionizing radiation, such as that found in X-rays and gamma rays, causes the formation of charged atoms, or ions, that can react with nucleotides and damage the DNA. UV radiation causes neighboring thymine bases to form chemical crosslinks, called thymidine dimers. These dimers must be removed from the DNA in order to properly transcribe or replicate the genes that are damaged by the UV radiation. Bacteria have specialized repair enzymes dedicated to the removal of thymidine dimers. However, if the amount of UV exposure is too high and the damage is extensive, the cell cannot repair the damage and cannot replicate or survive. This is why we can use UV light to sanitize instruments in hospitals, doctors' offices, and dental clinics. In addition, the genetic material of an organism can be altered through horizontal gene transfer and recombination. In horizontal gene transfer, genes are transferred between cells of the same generation. A portion of DNA from a donor cell is obtained by a recipient cell. This piece of DNA may be incorporated into the recipient's DNA through recombination. Recombination occurs when pieces of the donor DNA are exchanged with pieces of the recipient's DNA. The simplest path to recombination occurs through genetic transformation, in which naked pieces of DNA are taken up from the environment and incorporated into the genome of the cell. Transformation was discovered during studies to develop a vaccine against Streptococcus pneumoniae, a cause of bacterial pneumonia. The encapsulated form of this organism is pathogenic, and it will kill mice if injected. A non-encapsulated strain, however, does not cause disease in mice. Researchers were experimenting with heat killing the encapsulated strain and then using the dead microorganisms as a vaccine. If the heat-killed bacteria were injected into a healthy mouse, it survived and remained healthy. However, if the heat-killed encapsulated organisms were mixed with live, non-encapsulated strains, the mouse then died. It turns out that although the heat-killed bacteria were dead, their DNA was relatively intact. The living, unencapsulated bacteria that were mixed with the heat-killed cells picked up the genes for capsule formation through transformation and recombination, turning a non-pathogenic organism into a pathogenic one. Another way in which DNA can be exchanged between bacterial cells is through conjugation. In this process, a sex pilus is used to bring two cells close to one another, and DNA is exchanged across a bridge in the membranes. Like transformation, the new DNA can be incorporated into the genome of the cell. A third method of gene transfer in bacteria is transduction. In transduction, a virus that infects bacterial cells, called a bacteriophage, transfers DNA from one organism to another. Think back to our lecture on viruses. During the viral life cycle, viral DNA is copied and packaged into new viral particles inside the host cell. Remember, bacteria do not have nuclei to protect their genetic material, and their DNA is exposed in the cytoplasm. Pieces of bacterial DNA can be packaged into the new bacteriophage instead of the viral DNA. The virus containing the bacterial DNA is released along with the other viruses and can then transfer that DNA to a new host cell, where it can be incorporated into the genome. Plasmids are small circular pieces of DNA. They may carry genes for specialized processes or for survival under certain environmental conditions. A conjugative plasmid is required for conjugation and carries the genes for the formation of the sex pili. Dissimilation plasmids carry genes for the breakdown of unique sugars and hydrocarbons. R factors are plasmids that carry genes for antibiotic resistance. These plasmids are copied along with the chromosome during replication and are passed from one generation to the next. They can also be passed through horizontal gene transfer, through transformation, conjugation, and even transduction. Transposons are small segments of DNA that can move from one region of DNA to another. If they land in the middle of a gene, they can disrupt its function. Each transposon contains the genetic information for their movement, the simplest of which contain a gene for the enzyme transposase, which cuts and reseals DNA, flanked by recognition sites called inverted repeats. Complex transposons carry other genes, such as those for antibiotic resistance. Transposons can be carried on plasmids or viruses, and they can be spread from one organism to another. In this unit, we discuss the principles of microbial genetics. The genes of an organism determine its characteristics. Because they reproduce so rapidly, the characteristics of bacterial populations are constantly changing. Mutation and recombination provide the raw material for evolution. Evolution is defined as changes in the genetic makeup of a population over time. Organisms with the best characteristics are the ones that are most likely to survive. As we begin to look at the health implications of various microorganisms, it is important for us to remember that every medical intervention can provide selective pressures that can encourage proliferation of certain traits within a bacterial population. Antibiotic resistance is a prime example. This concludes the unit on microbial genetics.